Today's video lecture topic is going to be the cell cycle, the process of how cells grow and divide and multiply in number. Focusing on the mitosis cell cycle today. A separate video will focus on the meiosis cell cycle. We're also going to talk about how the mitosis cell cycle is connected to cancer. So as always, please make sure you're filling in your video notes organizer as you watch the video. Pause, rewatch as many times as you need to because this is a complicated topic with a lot of memorization, a lot of tiny little details. So don't skip out on this one. Okay, so first of all, let's talk about the difference between unicellular and multicellular cell reproduction. In a unicellular organism, it's really simple. You just duplicate the DNA and then split the cell in half, and essentially splitting the organism in half, and you're done. And that's a process called binary fission. It's an asexual reproduction process. In multicellular organisms, you actually have to divide the individual cells so that the multicellular organism can grow, can repair injuries, whatever. And that process is called mitosis, and that's what we're going to focus on today, the cell cycle mitosis process. So now here's the problem. As a cell gets larger, right, it's taking in nutrients, it's getting bigger, its volume actually increases a lot faster than its surface area. And the problem with that is you're increasing the inside so much, but you're not increasing the outside so much, and the outside can only take in so many things or let out so much waste that it cannot supply itself with nutrients or expel the waste that it needs to anymore once it gets so big to a certain point. And you can see that ratio of surface area to volume here. So while the inside of the cell, the volume is getting really big, the actual surface area hasn't really changed all that much. It's gone from 1 to 2 to 4 micrometers. So we have to have this process of making more cells. So that is what cell division is. Cell division keeps the cell from getting too large to where it can no longer support itself. It's also the way that the cell reproduces so that you can grow. You have to make more cells in order to be able to get larger. And also so that you can replace cells that have been injured, so that you can heal injuries. So it's for growth and repair, the mitosis cell cycle. The cell cycle is the series of events in which a cell duplicates its DNA and all of its organelles, its contents inside, and then divides to produce 2 new identical daughter cells. So the goal of mitosis is to make more of the same cells, which makes sense, right? If you want to repair injured skin cells, you want to be able to make more identical skin cells. So the purpose of mitosis is to make more identical cells, more of the same cells. Now here's the thing, cells actually divide at various rates, at different rates, which becomes a problem with things like liver cells. So liver cells divide only every year, every couple of years. They have a very, very long cell cycle. So this is why doing damage to your liver is such a problem. So we know that you can induce liver damage by drinking too much alcohol. Well, the problem is that when you damage those liver cells, their cell cycle process is so long that they cannot replace themselves at a quick enough rate, which is why you end up with things like cirrhosis of the liver. Some of your cells, like your nerve cells, your neurons, will never divide in your entire lifetime. So you don't want to damage your brain cells because they're not going to come back. They do have a cell cycle, but it's so long that they're never going to reproduce during your lifetime. Now, on the other hand, the cells, the epithelial cells lining your gut, divide more than once a day. Okay, their cell cycle process is only a couple of hours, which makes sense when you think about where they are. They're in a really sort of hostile environment, right? Your intestines, your stomach. So their cell cycle is going to be very quick. Okay, so now we're going to talk about the details of the cell cycle process. So I want you to memorize and write this down somewhere, this little phrase, IPMAT-C. I stands for interphase. This is where the cell is going to spend most of its time. 90% of a cell's life is spent in interphase. This is where the cell is growing, carrying out normal cell functions, and replicating its DNA. And then the PMAT, IPMAT-C. The PMAT is the mitosis portion of the cell cycle, prophase, metaphase, anaphase, and telophase. And this is where a cell is actually going to divide its contents, to divide its nucleus. And then IPMAT-C, so I was interphase, PMAT was mitosis, C is cytokinesis. This is the actual cutting of the cytoplasm, which is where the new cell is created. So let's get into these details here, starting with interphase. So you can see G1, S, and G2 are all parts of interphase. Okay, this is where a cell spends 90% of its time. Mitosis is fairly short. Cytokinesis is even shorter. So let's talk about interphase here. First of all, make sure you copy down that diagram under number 7, writing those details under G1, S, and G2. So the first stage of interphase is called G1. This is where the cell is growing and just carrying out normal cell functions. It's taking in nutrients, it's letting out waste, doing its normal cell thing. The S stage of interphase, S stands for synthesis. So the thing being synthesized in the S stage is DNA. This is where the DNA is replicated so that the cell can split. You have to replicate the DNA. If you want to start with 46 chromosomes and you want to end with 46 chromosomes, you have to duplicate the DNA first so that when you divide the cell, you're ending up with the same thing that you started with. So that happens during the S stage of interphase. And then the G2 stage, or the gap2 stage of interphase, is our checkpoint stage. This is where the cell is going to do its final checkpoints to make sure that it is ready to divide. If you have made mistakes in the duplicating of your DNA, you don't want to divide that cell. So that checkpoint, to make sure that everything has gone right, is going to happen in the G2 stage of interphase. So all of those stages, growing, carrying out normal cell functions, duplicating DNA, and checkpoints, those are all really, really important. So the cell is going to spend a lot of time making sure that they happen correctly. Okay, so we're going to use the term chromosome a lot. So I want you to make sure you understand what a chromosome is. A chromosome is a carrier of genetic material, DNA, that is found in the nucleus. And this information has to be copied in order to be passed on to future generations of cells. So a chromosome typically is this X-like structure that is made up of two sister chromatids. So one half of the chromosome is a chromatid. So here's a chromatid, here's a chromatid. Those chromatids are held together by centromeres. So normally in interphase, DNA exists in this double helix form, what we're used to seeing. When it actually condenses to form a chromosome, that happens fairly quickly. Most of the time, your DNA is not in chromosome form, even though we see that little X picture a lot. So you can see on my board here in class, I have the chromatin condensing into a chromosome. So DNA normally exists in this long winding structure, the double helix form, that is called chromatin, which condenses into the chromosome before the cell divides. And remember, the chromosome is two chromatids held together by a centromere. Okay, that's really important, so make sure you understand that. Make sure you have all that labeled. Pause and rewatch that if you need to. Okay, so here's our chromosome, which is our X structure, held together by a centromere, holding together the two sister chromatids. So, in all of the cells in your body, except for one, that's your sex cells, every other cell has 46 chromosomes, 23 pairs of chromosomes. You've probably heard that before. I'm going to use a term called diploid, 23 pairs. Diploid means two pairs of chromosomes. Two sets of chromosomes. So 23 pairs are two sets of chromosomes. That's in every cell in your body except for your sex cells. Your sex cells only have one set, so they're haploid. They have half the number of chromosomes. So in your sex cells, your egg or your sperm, depending on if you're male or female, you have 23 chromosomes in those cells. Now think about why that is. When a mommy and daddy love each other, right, the eggy and the spermy come together. So the egg has to carry 23 chromosomes, the sperm has to carry 23 chromosomes so that when they come together during fertilization, that chromosome number is restored to 46. And now that baby is starting with 46 chromosomes and that cell is going to start going through mitosis. But every other cell in your body, except for your egg or your sperm, have 46 chromosomes in them, or 23 pairs of chromosomes. Those replicated chromosomes that are being bound together are called sister chromatids. Now this is really important because eventually those duplicated chromosomes, those duplicated chromatids, are going to separate during anaphase. So make sure you understand the difference between chromatid and chromosome. You've probably seen this map before. It's called a karyotype. It's essentially a map of somebody's chromosomes. Now remember, DNA doesn't always exist in this form. DNA is usually in its double helix chromatin form. But when the chromatin condenses into chromosomes, you can take a picture of it and you can map it out, and that's called a karyotype. So you can learn a lot about a person by looking at their karyotype. For example, this would be the karyotype of a female because you have two X chromosomes, and you can see that, this is in 23 pairs of chromosomes. It's normal. By looking at a karyotype of someone, you can tell if they have certain genetic disorders. For example, if they have 3 13s, then you would call that trisomy 13. Okay, that's a genetic disorder. So this is a karyotype, a map of someone's chromosomes. So we talked a little bit about how in that G2 phase, we have our checkpoints. Those checkpoints are two things working together, cyclins and CDKs. And these are, enzymes and proteins that are working together to tell the cell whether or not it should divide. So at all of these little checkpoints, it checks to make sure, okay, was everything done correctly? And if the answer is yes, then it sends a signal to the cell that says, okay, keep going through the cell cycle process. But if something has gone wrong, for example, if the DNA has not replicated correctly, then at that checkpoint, the cell is going to say, you know what, we made a mistake. We don't want to divide this cell. And we're going to talk about the process of what happens if there is a mistake in just a second. But we're going to assume that everything has gone right, and we're going to go through the different stages of the mitosis cell cycle. So remember, you have I, P, MET, C. Write that down. Interphase, then the stages of mitosis, prophase, metaphase, anaphase, telophase, and then cytokinesis, I, P, MET, C. So we already talked about interphase. Now let's get into our stages of mitosis, prophase, metaphase, anaphase, and telophase. In prophase, and you're going to want to draw these pictures and tell me what's happening during each stage here for number 16. Take some time to pause on this if you need to. The chromatin, that double helix form, is going to condense into chromosomes, and the nucleus is going to disappear. You don't want that membrane surrounding the nucleus because you need those chromosomes to move all along the cell so that the cell can divide. These little structures called centrioles are going to form, and they're going to start producing these little fibers called spindle fibers, and you're going to see why those are really important in just a second. Now, this is the longest phase of mitosis, but remember, not the longest stage of the cell cycle. That's interphase. But it does take some time for the chromosomes to condense, to form, for the nucleus to disappear, for the spindle fibers to start to form. Now, metaphase happens pretty quickly. All of a sudden, those chromosomes are going to attach to the spindle fibers, and they're going to line up along the middle of the cell. So remember, metaphase, think M, meta, middle. Chromosomes are going to line up along the equator of the cell. Those spindle fibers are going to attach to the centromeres. Okay, so that's metaphase. Pause on that picture. Here's what it looks like in real life. You saw this in the microscope. And then in anaphase, those spindle fibers are going to shorten, and they're literally going to pull apart those chromosomes back into the sister chromatids, and the chromatids are going to head to the opposite poles of the cell. So they pull the chromosomes apart, and those chromatids are now going to be pulled to the poles of the cell. You can see what that looks like here in real life. So they line up along the middle in metaphase, and then in anaphase, they get split apart and pulled to the poles. In telophase, those chromosomes are going to unwind back into chromatin. The nucleus is going to reform. Now, in animal cells, the cell membrane is going to start to pinch in here. That's not going to happen in plant cells. In cytokinesis, the cytoplasm is going to be cut. So now you're actually going to have two cells. In animal cell, remember the cell membrane started to pinch in? It forms what's called a cleavage furrow, and then that's going to pinch all the way in cytokinesis so that you end up with two complete daughter cells. Now in plants, you're not going to have a cleavage furrow. You're going to have a cell plate start to form in between the two cells in cytokinesis, and that's going to be what becomes your cell wall. That's why plant cells look like little bricks, because they have a cell plate that forms and becomes the cell wall. Okay, so again, there's animal cell, the cleavage furrow starts to form, the cell membrane pinches in, completely pinches apart during cytokinesis. In a plant cell, you have the cell plate start to form, and that's going to be what becomes the cell wall. Okay, so I'm going to show you just some pictures of what these look like in real life. That's what the cleavage furrow looks like in an animal cell. That's what the cell plate looks like in a plant cell. And here's all the different stages, okay? Interphase, prophase, metaphase, anaphase, telophase, cytokinesis in an animal cell, interphase, prophase, metaphase, anaphase, telophase, cytokinesis in a plant cell. All right, now cancer. How does cancer relate to mitosis? Well, cancer is actually uncontrolled mitosis, the uncontrolled growth and division of cells. So it's mitosis gone wrong. Remember we have all those checkpoints? This is when the checkpoints aren't working and they're sending a signal to the cell that says, yeah, divide, 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 divide. And they never send a signal to stop. Now, growing more of the same cells isn't necessarily the problem. The problem is that those cells, those cancerous cells that are reproducing, your body is going to supply those cells with nutrients and food, which means they're not supplying the normal functioning cells with the nutrients and food that they need. That is the problem, that your body is supplying cancerous cells that can now crowd out the normal cells, resulting in a loss of tissue function. So the changes that occur in the regulation of cell growth and division of cancer cells are due to what's called mutations, and you're going to hear that term a lot. Carcinogens are substances that can cause mutations that lead to cancer. So for example, we know that there are those toxic chemicals in cigarette smoke that are carcinogens. They're carcinogenic, meaning that they can cause cancer. There's lots of research to prove that. So they would send a signal to a cell to divide. That's going to form what's called a tumor. And the problem is that your body is going to supply those cells instead of your normal cells, your normal functioning cells, with the nutrients that they need. Okay, so what happens if something goes wrong? Well, luckily, your body is actually capable of, at those checkpoints, saying, hey, something has gone wrong, and then signaling to the cell to program a cell death. So apoptosis is programmed cell death. In other words, it's cell suicide. Now, sometimes this happens naturally. For example, in the fall, there are little cells that attach the leaf to the tree, and those go through apoptosis to let go of the leaves so that the tree is not wasting its time supplying those leaves with nutrients when water is less available, like in the wintertime. This happens normally in a baby, right, in development. You actually have skin cells in between all your fingers and all your toes that go through apoptosis. That's why we don't have them anymore. And hopefully this is what occurs when regulators sense that something has gone wrong. So here's the pathway that I want you to draw for number 21. So here's a healthy cell, something goes wrong, and the cell has two options. It can either repair those errors, or those errors can remain, the cell is unable to repair those errors, and then the cell goes through apoptosis. That's the sort of pathway that can follow if there's a mistake that's made during the cell cycle. All right, so make sure you have all of your questions answered. Look up anything you need to, pause, rewatch anything you need to. Like I said, this is a complicated topic. Have a good day.